Welcome everyone to another episode in our series on innovating in rare disease research. I'm Duncan McGeechy. I'm the chair of the Prader-Willi Research Foundation of Australia, and we're launching this series in order to bring a little bit more information about some of the incredible cutting edge work that's being done to find new therapies and solutions for families that are living with a whole range of rare diseases. And obviously for us, we're particularly interested in people living with Prader-Willi syndrome. So it's excellent that today I'm very lucky to be joined by Dr. Tien. He's the CEO of Aardvark Therapeutics, which is doing some very, very interesting work in hyperphagia and Prader-Willi and other rare diseases at the moment. We're going to have a chat about that today. So what I'd like to do before we get into that is just let you know that today's conversation is educational content only, and you should always consult your healthcare provider about any kind of treatment decisions that you might be making. But having said that, we hope that you enjoy hearing from my guest today, Dr. Tien. So with that, welcome, Dr. Lee. How are you doing today?
Well, excellent. Thank you very much for having me.
It's a pleasure. Now, I'm going to dive straight in and we'll dispense with any small talk if that's okay. I was wondering if you might be able to let me know how your journey with Prader-Willi began and how that fits in the context of your life and career working in the clinical research space.
Okay, great. Thank you very much. Yeah, it was actually quite serendipitous. I'm a physician first and foremost. I went to medical school with actually ostensibly a goal to be a clinician. And over the years, I've actually been fortunate enough to be a founder or co-founder of several biotech companies, often contributing to the foundational science. And the exploration for the current drug we're developing as a lead program was really started as a curiosity. I was focused on revelations that the bitter taste receptor, that what you perceive as bitter taste in the mouth, those receptors are expressed almost universally among all vertebrate animals. That seems to be a universal feature of biology for vertebrates and mammals. And the bitter taste receptor, in fact, exists not just in your oral cavity, but throughout the body. It was fascinating by itself. And that there was observations that bitter foods lead to abrogation of eating behavior, proportional to how bitter something is. And it was just giving rise to a thesis of whether a bitter compound targeting regions of the body outside the mouth, so you wouldn't perceive something extraordinarily bitter, whether that could convey modification of eating behavior. So that was really initiated by just a scientific curiosity. One thing led to another. We had a lead compound. We saw really interesting results, and we elucidated that what was happening was that we were turning off eating behavior in a very different way than other drugs that people tend to think of for obesity, for instance. And the way the dichotomy that we observed is appetite versus hunger, appetite being a reward-based drive, hunger being a kind of a penalty or pain avoidance drive. And the motivations for eating are actually different. If you like something delicious, you eat something appetizing. And just to give you an example, we think that the GLP-1 drugs are primarily anti-appetite. Mice prefer chocolate to regular feed. You give them a GLP-1, they'll eat less, but they also stop caring to work harder to get chocolate, right? So it's almost like diminishing the appeal. But ours was working differently. It's taking away the penalty of fasting and so forth. And then it was doing this by way of gut-brain signaling. Well, long story short, we were trying to find out, once we figure out this prospective drug, what medical condition matched the mechanism of our drug the best. So we found the solution first, or the proposed solution, and then we're trying to find the matching medical issue that would pair along with it. And Prader-Willi syndrome actually fit the mold for a hunger-driven type of hyperphagia. And as we know, Prader-Willi syndrome individuals have more than just the hyperphagia. There's oftentimes anxiety, inflammation, very delayed gut transit times, and all those relate to the gut-brain signaling and the pathway that we believed our drug was activating, and that's why it made sense to delve in that direction. So that's how it worked, kind of backwards in a way from the scientific discovery. So sorry if that was a long-winded answer, but I just want to give you a
brilliant. That was fantastic. There's a lot to dig in there. Just to clean something up along the way, for anyone not familiar, just to clarify, when you talk about GLP-1s, you're talking about some very famous compounds that most people will be aware of. But can you just explain what the GLP-1s are? And just, I'm interested a little bit in the difference between the appetite pathway to managing, I guess, consumption, and the hunger pathway. If you could just talk a little bit more about what you mean by GLP-1s, and then the difference between those two approaches.
Okay, sure, of course. So GLP-1s are very well-known drugs now, like Novo Nordisk and Lilly have drugs, like Wegovy, tirzepatide, Zepbound. These are names that some people might be familiar with, that are injectable drugs, and they emulate a gut peptide hormone called GLP-1. So let me just rewind a little bit. When we eat food, food activates a number of responses that elicit a secretion of a symphony of gut peptide hormones to regulate eating behavior. Among that symphony of gut peptide hormones is something called GLP-1, which works both locally and systemically in the brain to kind of turn off appetite. And I guess the subjective experience is that, I guess, there's a diminished inclination to eat food, and people can lose weight at the appropriate dose, but there's also an expectation of nausea that occurs in a percentage of individuals, and that's one of the many gut peptide hormones that is released. We believe that our drug is stimulating the enteroendocrine cells that release that spectrum or symphony of gut peptide hormones, which may include GLP-1, but very importantly, it also elicits the secretion of CCK and other gut peptide hormones. And those are acting locally at the vagal nerve, which is really the connection between the gut and the brain. And those might be familiar with the vagus nerve. It's a very important communication between the gut and the brain. There's actually a two-way communication. What we believe we're doing is activating the afferent, or the gut to the brain traffic, not the brain to the gut. And that selective ability to turn off hunger is really what differentiates that. So it's like a different set of gut peptide hormones that are being elicited rather than the GLP-1. And just one more layer of refinement, just to clarify. So we release natural levels of GLP-1 in order to turn off appetite, and these are very minute amounts of natural compound that gets rapidly degraded. The ones that are made by the pharmaceutical companies are made to not degrade as quickly, because those usually rapidly made and degraded. So the artificial version lasts longer, and that's what that does. And our drug, believe, is basically inducing the natural secretion of these gut peptide hormones.
And so, are there instances in the wild, in sort of the natural functioning of the human body, where the bitter or the bitterness receptors that you're seeking to target or activate occur naturally, or what are the circumstances? Is that part of when you're full? Does bitterness receptors get triggered, or is it only when certain types of substances come into contact with those receptors and the body then reacts?
So that's great. There's actually multiple switches and certain redundancies in the body. Food can activate enteroendocrine cells to release these gut peptide hormones. We believe that bitter taste receptors are almost could be thought of as a secondary pathway to also induce the release of these same gut peptide hormones. So you can activate them with food or with a bitter substance. And there's some, empiric evidence that, people who drink coffee, the more coffee that is drunk as a population, there's some observed proportional weight loss, and it's not always due to the caffeine, which is kind of almost reinforcing that point. And activating the bitterness is just another way to enhance that expression. And we believe that perhaps in Prader-Willi syndrome, the patients don't get normal satiety with food because the normal pathway of food activation may be diminished or not as effective. So we believe amplifying that with that secondary engagement is basically compensating for that lack of normal feedback.
Okay, so I'm just going to jump ahead a second. It's interesting when you're talking about, using whatever tools are available within the human system to achieve an outcome. Then obviously you're talking about some innovative research. And once we get to the clinical stage, can you talk a little bit about how Aardvark specifically, but the industry in general, manages patient safety as you start to move through the research stages to see what's possible with some of these approaches?
Of course, safety is, of course, of primary concern. No one ever wants to inadvertently introduce harm to a patient. We're all trying to do productive, favorable things for the patients. And there are many layers of redundancies to help ensure that something is safe. So, of course, there's a lot of ways to chemically assess something even before you get into animals, whether it looks like it could be potentially dangerous or whether it has a structure that's similar to something that else has caused problems. Then you test it in animals in more than one species just to be sure, because animals are not humans. So then, even then, you don't have 100% assurance that there's not something, because you don't know what you don't know, right? So you still have to be open to identifying prospective dangers. You try to identify what are the first warning signs of the toxicities you would expect, because things with ever increasing dose, that there's either a manifestation of one type of maybe it's nausea, maybe something else that would be the first thing, and this is something you try to get some insight into in animals. And then when you go into humans, there's a very careful increased dosing that's generally observed. And then there's a very comprehensive panel of observations. And of course, even as you continue to assess the drug in larger trials, you want to make sure that there are not some rare occurrences of things that could not be foreseen. So we're always trying to reduce as much as possible, but it's always a working process.
I understand. So in that context, then I'm interested, what were some of the key milestones for you and your team in the progress up to the point you're at now with this particular approach to hyperphagia?
There were many insights. So I can identify scientifically, our drug has some interesting characteristics that are not typical. Like one of the features is that less than 1% of the drug is absorbed in the bloodstream, which is normally not a favorable feature for a drug, as normally the intent is to make it systemic. But in our case, the drug is targeting the receptors in the gut. So we didn't know that. We were actually ignorant of that fact. And then it turned out to be one of the better parts of this drug was the fact that it was gut restricted. And then by kind of being curiously looking at how this could be working, despite the fact that it wasn't really systemically distributed, we then elucidated this gut-brain signaling. So that was actually a very big aha moment and gave us a lot of inspiration and a lot of assurance. Reflecting back on the safety concern, I mean, you never want to be too cavalier about it, but the fact that it was so gut restricted gave us additional confidence that we had a big safety margin. And then we started testing it in different conditions. And then I think we're very encouraged by phase 2 data, and this is what kind of gives us encouragement to keep proceeding. So we want to be good scientists and good clinicians and stewards of patient care. So we're very encouraged by all the information that we keep on getting back.
So I just want to pause and just clarify on that point. And I always find it beneficial when I'm happy to be the most ignorant person in the room and I can ask these questions. When you talk about 1% absorption into the blood, does that mean only a tiny amount of the drug that's ingested, it goes from the gut into the bloodstream? And in this case, the rest of the drug is passed out of the system without being absorbed into the body, and that small amount that's absorbed into the bloodstream is sufficient to do the work that you're hoping the compound will do? Is that a correct understanding, or
almost there, except that we don't think that the amount that escapes the gut and goes into the general circulation, that's not as important. I mean, we think that the real effect is the fact that the drug is remaining in the gut. So it's doing what it needs to do as it's transiting the gut. So not to be too, I hope it's not, meal time, but, through number two, you just pass through the body, but it's activating those receptors as it's moving down the gut.
Okay, I really appreciate that. That's a good clarification. So, one of the topics that we're exploring at the Prader-Willi Research Foundation of Australia in our community is the beneficial and necessary role that patients and families can have in this research. And I'm interested from your perspective, what do you see as the role of patient and family input into your work and into this area of innovative research in rare diseases?
Oh, there are many dimensions in which this was actually very critical. One of the key components is to have actual clinical insight into what's important. I mean, I recalled Prader-Willi syndrome in medical school, but and hyperphagia is of course kind of a key element. But the anxiety, the importance of the inflammation and the constipation and other clinical features is very important to kind of understand holistically all the challenges, and then it gives us insight to and appreciate what's the best approach to ameliorate these things, and then also even as we are ultimately trying to convey benefit for patients, we also want to make sure that we're measuring the right things, and we're also balancing all the considerations, and also making sure we're being responsible in picking up good signals and being wary of things that we should be worried about, and even in the conduct of trials, things that are easily overlooked, even if clinicians can be very experienced, but there's certain challenges that families with Prader-Willi individuals face in terms of travel and logistics, and those things are hard to appreciate unless you've had interactions with individuals.
Look, I mean, one of the fascinating things about research into Prader-Willi, from what I can see, is our progressive understanding of how the different manifestations of Prader-Willi syndrome, which can be unique to an individual and are different in every single case, how they kind of fit together and what that tells us about the nature of the human body. And it was interesting earlier when you talked about the revelation of the gut-brain communication and signaling and the role of the vagus nerve. I was just wondering if you could take us back to before you had that insight and that revelation along the way. What was the kind of alternative assumption you made about how your compound might work in the body? What's the different way to think about how the body works alongside the gut-brain communication, which has become such a big topic in a lot of different areas?
Sure. So a priori, the general expectation is the drug needs to be systemic, and so for something that was affecting eating behavior, it's almost assumed, at least as a base assumption, that it needs to work on the brain directly, right? So it has to somehow get into your blood and bathe in your blood and then penetrate the brain regions in order to hit the neurons to affect eating behavior. In fact, that probably would have been my base assumption as well, frankly. And it was very not immediately clear, why our drug was working this way. And I think even this gut-brain signal, even though it's of increasing focus, I guess I'm still old enough that in medical school that wasn't even a thing when I was training. It was a
Is
very
that right?
So it's very rudimentary, and in fact, most of the attention was on the brain-gut connection, not the other way.
Well, okay. So now we're in a world where we're trying to activate other pre-existing systems in the body, which they handle the communication with the brain rather than the compound directly.
That's right. We're basically, leveraging the body's own relays to produce a therapeutic effect instead of trying to to manipulate it more downstream. So that's the approach that we've taken.
Yeah, that's really interesting. So just taking a step back and changing direction for a second, is this the first work you've done with a significant focus on a rare disease, or have you worked in this space before? And based on your experience, what are some of the unique challenges and maybe even opportunities from working in the rare disease space?
It's not my first rare disease program. I mean, but before I had an oncology program that I explored for a rare melanoma variant, and I think I think there's different scientists that approach things differently. It's very productive that people have different ways to investigate problems. But I've generally tried to work backwards from scientific first principles, and trying to understand a puzzle of biology, and then try to find the matching piece So this is really it wasn't just an intellectual exercise. I mean, I wanted to go into this because I thought there was a prospect of making a kind of a magnified impact if you could get insight into a disease process. And I think finding a solution to a rare disease is in lots of ways very elegant and satisfying, because then you have a bespoke treatment for a certain condition that's optimized. And although there might be expanded use cases for a drug, just selecting the correct patient population, seems giving a little level of scientific precision that is my general inclination.
Can you explain that a little bit about selecting the correct size or the correct population? What does that mean, and what does getting that wrong look like?
Well, generally speaking, it's every therapeutic is like a financial decision in which there's a risk-benefit assessment that one takes, and you generally have prospective benefit versus possible complications from a treatment, and that calculus is important in managing this. I mean, you give antibiotics, for instance, and everything you take has a countervailing side. If a drug is correctly chosen, that ratio skews more favorably to the benefit versus risk side of the equation, right? So that's just kind of like the approach. And if something could be broadly applied with still a favorable risk benefit to other conditions, I would always want to start with something that really has the most selective advantage. So if that makes sense.
No, that's really helpful understanding. It all helps us understand the way the world looks to the people that are investing time and effort and resources in this kind of innovative research. I think that's really helpful. Just on that, one of the principles of the Prader-Willi Research Foundation of Australia is that there are aspects to Prader-Willi syndrome from a research perspective that have potentially broader application. So obviously we're focused on people living with Prader-Willi. That's our laser focus. But in the real world, in order to draw investment and support and attention and collaboration, the prospect that a breakthrough in Prader-Willi might have broader relevance to the community, whether it's in obesity or mental health or other areas, is a benefit we see in terms of attracting people to working in this space. And I just wondered whether in the process of your work, you've had any sense of the possibility of addressing a particular problem for the Prader-Willi community might lay the foundation for significant insight and benefit to the broader population outside of our community.
Yes. Okay. This actually reflects back on what you asked previously in terms of why rare diseases is at least makes sense in a way first. So if we go back to the construct of appetite versus hunger, if someone with general obesity, for instance, there actually is, like in Prader-Willi, a very wide array of drivers for for nature wanting you to eat more than you want to eat, right? And sometimes it's maybe I'm just being a little bit facetious, but maybe 80% appetite, 20% hunger for some people. Sometimes it's 50-50. I would say in Prader-Willi syndrome, it's nearly 100% hunger driven most of the time. So if something is really great as a anti-hunger mechanism, it would make sense to in a Prader-Willi type of situation. But it doesn't mean that even someone with 20% hunger still has that remaining as a challenge, even if they take a GLP-1 and they take care of their appetite. Still have that hunger driver, which then comes. So even for general obesity, there is a prospective use, but it also is dependent on the relative contribution, but it would be complementary to GLP-1s. The other application that we've contemplated in the past is as an anti-inflammatory agent. I think before we've disclosed data on regulation of inflammation, and and Prader-Willi individuals have oftentimes a lot of inflammation, but if this works the way we hope it does and we expect it to, then there are other inflammatory or autoimmune conditions that could potentially benefit from expanded application in the future. So those are kind of like the ways that we gain insight to other applications. And then the other insight, by the way, is that we think that our drug is helpful for preserving lean body mass. So even as it is discouraging hunger, there's a push towards more lean body mass. We saw that this shift in increasing lean body mass and lower body fat percentage in a short period of time we dosed individuals in our phase 2. So again, there's other implications there for other indications, including general obesity.
Look, I don't want to take you outside of your area of expertise, but I feel like it'd be remiss of me if I didn't just explore this a little further while I've got you. The relationship between the hunger driver and the appetite driver and a potential misunderstanding of how that applies in different people. I just wonder whether, from what you've seen so far, has there been a traditional assumption of appetite as the main driver for hyperphagia in people with Prader-Willi? And from what you've seen, has that had an impact on, I guess, the way families try to think about how they should manage life with Prader-Willi and what the treatments and the options are?
Sure. I mean, I think the conversation about the distinction between appetite and hunger is not something that is often commonplace. We even our own theses on that difference is elucidated by our own work, right? So I don't think it's a common accepted dichotomy, but I do think that it really is a difference. I mean, if you could imagine if the world had only bland celery sticks for sustenance, there would be much less obesity in the world because most people wouldn't choose to eat too much of that. But if you have Prader-Willi syndrome, the drive to satisfy that hunger would still remain, and it wouldn't slow down someone with Prader-Willi syndrome as much as it would, someone that's driven by an appetite. So I think that difference is important in conceptualizing it. But I do think that there's probably lack of general focus on this dichotomy. But we hope to increase awareness of that, and we do think that definitely is a good way to think about things.
And I would assume that given the range of different parts of the human body and human experience that are affected by the gut-brain relationship, you talked about anxiety and other things alongside, hyperphagia, then I imagine that continuing to explore and understand this area of Prader-Willi and the human body is going to be relevant potentially to a whole bunch of different parts of life with Prader-Willi. So more work in this space, please, I think is the message from our community. Just to finish up, this has been a fantastic conversation so far, but I'm conscious of time. One of the sort of founding principles of the Prader-Willi Research Foundation in Australia is the need to build our capacity to do great research here. And in the process, what we've found as we've built that community is international and global collaboration is incredibly powerful. And I just wonder, from your perspective as someone, not from Australia, but looking to do work here, what do you see as the role of international collaboration and global collaboration? And what do you see as the beneficial role that Australia in particular can play in supporting and driving that kind of global research work?
That's fantastic. I mean, I do think that, especially in rare disease, but on almost any condition, a global collaboration really accelerates innovation and understanding. Everyone's different, and people in different countries have different phenotypes, and it's important when we actually pursue a therapeutic treatment that we contemplate all the variation in the human experience as well too. So that's very important, just to make sure we're comprehensive in our investigation and not neglect any the differences between individuals, but also even the different styles of management of a condition. We want to make sure that we're not being too unilateral in the way we approach things. We want to keep an open mind. I think Australia has been incredibly impressive to our colleagues, because I think the cohesion and the camaraderie and the communication and the awareness of prospective therapies and so forth is very inspiring. I think you're actually a model for a lot of places. I think Prader-Willi syndrome, unfortunately, is a issue that is spontaneously occurring in about 1 out of 15,000 births, and it's fairly uniform worldwide. And I would say Australia, there's a lot of awareness, a lot of excellent clinicians, and a lot of knowledgeable families. And I think this is, like I said, it's a great way to advocate for the patients in this region, but also a way to accelerate discovery and innovation for the rest of the world too.
Oh, that's fantastic. I'll take credit for all of that on behalf of Australia. So just to finish up, and I'm conscious that for regulatory reasons, we have to be very careful about straying into any discussion about some of the particular work that you're doing here at the moment. But having said that, I understand that you're doing a webinar on a particular piece of work that you've got going in Australia with the webinars with the foundation. I just wonder if you could just give people a little bit of insight into what you'll be discussing in that webinar, and then we can make sure it's available to anyone who's watching this.
Oh, sure. We were just discussing more about the reasons for us developing this program. We have a phase 3 clinical study, which we think that if successful, could potentially be the pivotal trial that would enable an approval of this medication for Prader-Willi. So we're enrolling currently. So we'll be discussing that and other aspects of our program, and going to further detail and address any outstanding questions relating to the rationale for everything.
So we can make sure that that information is available to anyone who wants to find it. Dr. Lee, this is the second time you and I have met. Both times it's fascinating, the work that you're doing and the insights you're generating, but it's also very exciting, the insights that you're generating and the potential for some of the work that you and Aardvark Therapeutics are doing. Thank you so much for joining us today. I think it's been really helpful for our community, and thank you for the work that you guys are doing.
I appreciate it, and I also don't want to take full credit for everything. I have an amazing team. They contribute so much. We couldn't do it without every person on the team, and we're going to try our best to introduce benefit to the community. So I appreciate the time and the venue, and look forward to our next conversation.
Brilliant. Thank you.
Thank you.
All right.
All right.
That was really good.
All right.
How did that feel? Did that feel good?
Yeah. I hope it was okay. I'm not as good as Corey.
No, from my end, it was brilliant. I mean, Kath will tell us if there was stuff we didn't cover or anything that we fumbled a bit. But no, I think that, I normally trust my own instincts. It was a really interesting conversation, and hopefully hopefully our community gets a lot out of it. I think this is really beneficial. I really appreciate you helping us with this.
No, thank you. I know I have to be restrictive in what I say, but I'm just very, very, very optimistic about how things will be in. And I think the whole team, we wouldn't be doing this unless we were believing in the larger mission. So I just wanted to convey my gratitude for you and trying to help, just facilitate awareness of what we're doing.
No, it's great for our community. So am I going to see you again? You're going to be down here over the next year or so?
Actually, I hope so. I think once things get going, if our chief medical officer, Dr. Mansi Jaman, she may be making a visit, and I would like to kind of join and say hi to some of the investigators and just see how things are going. I would love to go there again. I mean, I didn't see anything last time. I was just doing work.
You were in and out.
Yeah, I know it's a wonderful city. Yeah.